So the first thing I want to say is that um, a lot of this work wouldn't have been possible without support from the University of Southern California Sea Grant Program. Um, a lot of the work that I did was focused on the urban ocean, but also we did a lot of work on the islands. Today, I'm going to talk about some of that work that was performed in the 1990s to 2000 period, which is way back there for some of you guys. But I want to use my own personal experiences, perspectives, some of the literature, and some of the results that we got then to talk about historical changes in Southern California, rocky intertidal seaweeds and macroinvertebrates. I want to point out that we're going to emphasize the seaweed communities here, but you will see that we will be looking at some invertebrate work as well. So first, I think we all know that if we go back quite a ways and take a look at the LA area, that, things have changed a lot. This is an illustration of the LA Harbor way back in the early 1900s. And you can see that it doesn't look anything at all like this now. And I also think you all realize that the population in the Los Angeles basin has grown considerably. We have a lot of infrastructure that's put in place to handle our sewage and runoff of water after storms, um, and and so on. But all that wasn't there the entire time that our coastline was in this development stage of accommodating more and more humans. So there's something to be said about taking a look at what we did know and what we are now able to find when we go out and take a look at our rocky shore communities. So when we take a look at the historical records, a lot of what we know, and at least in terms of leading up to the 1970s, was based upon taxonomic studies and taxonomic records. So those taxonomic records are in the form of specimens that are in herbaria, species lists that were published, and monographs that outline the number of flora, the species that are in the flora, focusing on maybe brown seaweeds or red seaweeds or this area or that area. And the same really goes for the invertebrates. But I think what a lot of people don't know is that leading up into the 1970s, our ecological data, you know, gathered by standard ecological sampling techniques that generate not only species compositions, but also abundance data, they were extremely limited. We didn't know much in that ecological sense about what our rocky shore communities look like. And so when we started some of this work in the 70s, which I will report on, we didn't have much of anything available in a comparable, ecologically acceptable way to compare results that we were getting using standard ecological sampling techniques. Now, when we talk about taxonomic records, a lot of what we know in Southern California was generated for us by Nathaniel Gardner and William Albert Setchell, particularly Setchell. Gardner was a PhD student at UC Berkeley, who then took a position in the Berkeley Herbarium and worked hand in hand with Setchell on a lot of projects and studies. This is why when you look at the names of seaweeds today, you'll see a lot of them have credited Setchell and Gardner or Setchell with their original descriptions. Now, Setchell's records were in the form of exicata. These are essentially big, huge volumes where seaweeds are pressed on paper, identified, and then they were duplicated. And so they became lists of flora. as well as monographs that were written on the seaweeds of North America. Now, after Setchell and Gardner, we move into a period where folks, particularly in the Bay Area, Gilbert Smith and Isabella Abbott, George Hollenberg from Southern California, who collaborated with Isabella Abbott, began to digest and synthesize a lot of this information. And that led to the production of the Marine Algae of California, which is really the last published detailed book of what seaweeds are present in California. But you'll see Setchell and Gardner credited for a lot of the work in Southern California. Now, another guy who was really instrumental in developing our knowledge of seaweeds in the southern area, but also really in Pacific Mexico, was Yale Dawson. Now, Yale Dawson was mentored by Setchell at UC Berkeley. This guy was amazing. He was not only a prolific author, but he got his PhD in 2 years. He was amazing and just completely embedded in the issue of collecting data, principally specimens and taxonomic work, and then turning that immediately around in written publications. But one thing that Dawson did that was of value ecologically was in the period of between 1956 and 58, he surveyed 44 sites, rocky intertidal sites in Southern California. Now, he engaged in this project as part of an effort sponsored by the State Water Pollution Control Board, designed to try to get a handle on our water quality in the nearshore in Southern California, and particularly the issue of sewage impacts. Because at the time, sewage was being put into local waters off Palos Verdes, near LA Harbor at White's Point, and also in the area just north of Palos Verdes, now we call the Hyperion outfall. And a lot of that sewage was not treated or primary treated. And when you go outside from the urban Southern California, or you go down to Orange County and Laguna Beach, for example, or up to Ventura, the sewage outfalls were actually dumping sewage in the intertidal zone. So what really spurred the interest in trying to figure out what was going on here was the huge decline in kelp beds that occurred off the Palos Verdes Peninsula. This kelp bed off the Palos Verdes Peninsula prior to the 50s was as rich and expansive as the kelp beds off of Point Loma, but it had come down considerably to very little detectable kelp. And this was a big concern. Now, at the same time, by the way, there was a massive El Nino that occurred. And people didn't really understand what was really the causes for that kelp bed decline, but the Water Pollution Control Board made a massive effort to try to put all this together. Now, what Dawson did in surveying his 44 sites was he recorded the conspicuous species that were present, and then he made scaled estimates of their abundance. These are 42 of the 44 sites. As you can see, they're all on the mainland from up here near Government Point or Point Conception, all the way down here into San Diego near, this is Point Loma here. And it turns out that we decided in around 2000 to take a look at these sites here that were on the Palos Verdes Peninsula, and also these sites here in a separate effort that were distributed along Orange County. Now, we weren't the first to go back and look at Dawson's sites, but I want to point out, as you can see, that they're distributed throughout the Southern California region. There are holes here where there's a lot of sand here and here and here, and a lot of concentration up in here where there's a lot of little rocky outcrops. Okay, what I'm going to speak about today is almost entirely going to be about the mainland. Dawson did look at two sites on the islands, one on Santa Cruz and one here on Catalina. Okay, so what did he do? Well, he went out to these sites, and you have to remember that he was a taxonomist. This was his forte. And so he laid out a transect line. You can see a line here that has been put in place. And he walked along this transect line. He wandered over on both sides of the line and recorded the species present, made herbarium collections. And with his estimates, he used a 4-point scale. Now, there's no real logic for this scale. It's just what he chose. If the seaweed was absent, the species was absent, it got a 0. If he decided it was rare to scant, he gave it a 1. If it was occasional to frequent, he gave it a 3. And if it was really abundant, he gave it a 10. And so every site for every survey had scores for each of the species that were sampled. Now, his sites were resurveyed in 1969, 15 of them, in the LA area by Tom Whittison, who was working out of Cal State Long Beach. And by the way, before Tom was at Long Beach, he was at UCLA. And he basically tried to be Dawson. He visited all of his sites, used the same techniques that Dawson did, and used Dawson's abundance scale. Now, about 8 of those sites, mostly up in the Santa Barbara Channel area, were resurveyed in 69 to 70 by Nancy Nicholson and Bob Simberg. And they looked at 8 of Dawson's sites, and they were looking at the 1969 Santa Barbara oil spill. And we're trying to find data to compare what they were finding against what was known previously. And this is all they had. Now, Ronald Tom came along. He was a master's student with Tom Whittison, subsequently went on to take a position up in Washington State. And Tom resurveyed Dawson's mainland sites, all 42 of them. And he did the same thing. He did the same sort of Dawson approach. He used what I'm going to call a Dawson-like abundance scale. It was slightly different. And you can find his complete records in his master's thesis at Cal State Long Beach. In other words, the species he found at each of the mainland sites and what scores he gave them. Then lastly, Leslie Harris, who works at the LA County Museum, she's largely now a polychaete taxonomist and curator. She was working at SQUIRP at the time and took a look at Dawson's 5 sites on the Palos Verdes Peninsula. She has species lists, and she did a really detailed job of looking at even the smaller species. So her emphasis was on floral richness. And as you might imagine, when you try to look at floral richness, well, the eyes of and knowledge of the investigator, the details you use to do the sampling, what do you bring back to the lab to identify, they have big effects on how many little smaller inconspicuous things you find, and unless they found quite a few. Very, very thorough look. So in 2004, this is the time that this was published, one of my master's students, Amanda Gerard, did her thesis work by relooking at Dawson sites that were distributed along the Palos Verdes Peninsula. Now, we used what we hoped would be a technique that was more ecologically valid in terms of producing abundance data, but at the same time resembled, at least so we could make some comparison with Dawson's work. So we attempted to relocate his original transect line. He only had one at a site. We laid down a second one about 2 meters away, up coast or down coast, depending on the site, but directly parallel to Dawson's line. And then we used point contact techniques to score the seaweeds present at 2 centimeter intervals along the lines. That gave us a lot of points, and we used those to generate percent cover of the taxa. But we also did a search of the area of the site, similar to Dawson did, to create a complete list of the conspicuous species. So we were trying to generate data that we could compare with Dawson's studies. And we, with the conspicuous species that we found that were not in the point context, they were treated as a member of the flora and treated accordingly, given a very minimal, a default minimal cover value. Now, a little later, we had a class, a phycology class, and we made it a project to go look at Dawson's 4 Orange County sites. And we used the same basic methodologies and did thorough searches and brought specimens back and so on and so forth. Now, this is a digestion of these results. So first of all, I want to point out that relative abundance is not so easy to generate. So this is you have to take this as a scale that shows and allows comparisons of the 2 different kinds of work. Dawson's original work, and then the work that we did with Amanda Gerard. The surveys were actually done in 1998, and then the Orange County surveys in here. So for example, if we found a species was found as 10% of the total percent coverage at a site during 98-2000, we would give it a 10% on the relative abundance scale. And same with Dawson's. So we divided our species data into what we call types of seaweeds, or you can also refer to them as form groups. And I'm going to be talking quite a bit in this short period about form groups. But the first thing I want to point out is when we look at rockweed canopies, and these would be Sylvitia, or what was then known as Hesperophycus, but what is now known, I guess, as Fucus serenoides. These are upper shore rockweed canopies. And if you take a look right away, you can see Dawson was finding these rockweed canopies in all the sites, pretty much, except for this one at White's Point. In 98-2000, we really only had the rockweed sites, rockweed cover here at Flat Rock on the Palos Verdes Peninsula. They were completely gone, a little bit here at Dana Point. So we would argue a big change in loss of rockweeds and rockweed canopies, largely driven by Sylvitia, which is by far the more abundant of the 2. On the other hand, if you look at crusts and algal turfs, well, you can see that Dawson found them to be pretty abundant. I mean, they were there pretty much abundant in all these sites. This is a characteristic of Southern California shores. But in our surveys in the 2000 period, we found these to be much higher at all of these sites. This has repercussions that I'll discuss later. And then these mid-size, what we often refer to as fleshy algae, these are largely red seaweeds that occur from the lower intertidal up through the mid-intertidal zones. You can see that Dawson found them to be pretty common parts of all of his sites here. On the other hand, we found them to be much less abundant in this period when we did our resurveys. So you can see that it looks like there are some pretty significant changes. If you look at these and break these algae up into form groups, this is one particular type of form group breakdown or categorization you can use, that crustose algae were increasing in abundance, articulated corallines were increasing in abundance, fleshy algae were decreasing. This, by the way, is the number of sites where we detected a decrease and the number of sites where we detected an increase. The kelps and fucoids were largely decreasing, coupled with no change. The sheet-like algae, like ulva and pyropia, anaeromorpha, they were decreasing. The filamentous algae were decreasing, and we found some increases in seagrasses, interestingly enough. So clearly some changes. So the conclusion here would be that if you look at these macrophyte floras, there were apparent decreases in the abundances of larger fleshy algae and apparent increases in the abundances of articulated corallines and crustose algae since Dawson's surveys. Now, I should also point out that Thomas Widdowson found a similar decrease in larger fleshy algae when he did his resurveys in 1969. And if you look at Setchell and Gardner's original herbarium specimens that they made prior to Dawson's work, you'll see that a lot of these fleshy algae, this is an example of one, Chondrocanthus, they were represented on herbarium records by pretty darn large specimens. You go to these sites today, and most of the specimens you're going to find are a lot smaller. Now, herbarium records are pretty weak evidence because most people go out when they collect specimens, they collect some really nice looking material. But I would argue from my own personal observations, experiences, and looking at the literature and our work with gathering data here in this period, that a lot of these changes that occurred probably took place prior to the 1950s. So they took place probably during the World War II population increase and urbanization boom. But they were continuing and ongoing as you move forward from Dawson's period into the current period, which at the time was in the 2000s. Okay, in the 1970s, what was then called the Bureau of Land Management and is now the Bureau of Ocean Energy Management sponsored a big program to determine the status of biological communities in the Southern California Bight. This work was being sponsored and it went on for 3 years, and it was being done in order to pave the way for offshore oil and gas leasing in federal waters, which was going to start up then in that late 70s period. The agency needed those data in order to write the environmental impact statements. They needed to know the status. And they also wanted to continue these surveys so that they would be able to modify or examine the effects of oil and gas exploration and extraction that followed. In other words, they would have a baseline to follow. It turned out that they never did any of the follow-up work that was designed to take place. But in the 70s, Mark Littler got this big contract, and myself and Roger Seepy, an invertebrate zoologist, we were co-investigators on this project. And this project was to make ecological assessments of the rocky intertidal macrophytes and macroinvertebrates, eventually at 22 Southern California sites. And this work was done using robust ecological sampling, and it was intensive work. So for each of the 22 sites, we wound up with species composition, species distributions, and their abundances. Here are the 22 sites that were sampled. Now, I want to point out right away, and I think you can see this, that there was a lot of heavy emphasis on the islands. Now, there are two reasons for that. One was some of the offshore oil and gas activity was going to take place more in the channel areas. And given the circulation patterns, might affect the flora and fauna and fishing, really, that was largely taking place here on the islands. But the second reason was that we didn't know anything much about the distributions and abundances, ecological abundances of based generated abundances of rocky intertidal macrophytes or macroinvertebrates on these islands. In fact, there was only going into the studies in the 70s about 3 papers you could even refer to. One on San Nicolas, which is out here, I'm sorry, and one on Santa Cruz, and then a big list of species that was collected off Catalina as a precursor to building the USC lab there. Now, on the mainland, there were several sites that were studied, but really the work concentrated on the islands. So in the period between 1994 and 2000, 1999 and 2004, we got funding from BLM or BOEM to look at 9 of these sites. The USC Sea Grant also provided funding for that work. And these are the sites that are generated in orange or listed in orange with the orange arrows. Now, today I'm going to talk a little bit about these mainland sites. And I want to immediately point out that 2 of these sites, this one at White's Point and this one at Corona del Mar, are really in the more urban part of the LA basin. This site here up at Paradise Cove in Malibu, up in the upper part of Santa Monica Bay, and this one here at Dana Point are a little more on the outside edges of urbanization. Not that they're not urban influenced, but they certainly don't receive the same degree of urban impact as these 2 sites did at the time. Okay, we're going to talk about these 4 sites now. So this is a result of looking at the community compositions, looking at sessile invertebrates and all of the macrophytes. This is cover data that was generated. And this is a non-metric multidimensional scaling plot. So each of these points that you see here, these triangles, represent or depicts the community composition based upon the cover values of macrophytes and sessile macroinvertebrates. So let's take and look at here at Corona. Let's look at White's Point. This is an easier one to look at. So there were 3 sampling periods in the 70s that were executed in the winter, in the month of February. And when we did our work at White's Point, we did that work in March. And so these 3 communities that were sampled in the 70s had about a 70% similarity in composition among them, which is indicated by the dotted outline here. Our study in the 50s, our study in 2000 had joined these others only at a 50% similarity level, which is indicated by this orange line. So that means that this White Point community looks to be quite a bit different clearly in its composition in 2000 compared to what it was in the 70s. This is Dana Point. Now we only have 2 samples at Dana Point. We got one done in the 70s and one in 2001. 50% difference. Again, a good bit of difference between the 2 sampling periods in terms of community composition. Now Malibu was a little tighter with the 2 sampling periods basically having a 70% similarity. And then Corona del Mar, a little more variable, with the samplings being done here having a 50% similarity overall in the 70s. But our 1999 sample at Corona del Mar jumped way up and tends to look a lot more like White's Point. So when you run a Permanova on this, which is a permutation test, you can see that the differences, the sites themselves are distinct. They're significantly different from one another at a highly significant level. The sampling periods are significantly different, and there's an interaction between site and year. That interaction is largely driven by this Corona del Mar shift. So we looked deeper into what was going on at these sites. We did a lot of different analyses, but the one I want to share with you today is to again look at these different form types of seaweeds. So let's look at Dana Point. The blue would be the average for the 70s. The red would be the period in 2000. Over here, we have fleshy algae. So these are these sort of largely red, some brown, lower intertidal to mid intertidal fleshy algae. These are larger seaweeds like lower intertidal kelps, like Icenia, Egregia, sargassums, and so on. These are articulated corallines, and this means they're the upright, erect corallines you would find like Corallina, Lithothrix, species like that. These are crustose algae. So these are like largely two-dimensional crusts that occur on rock surfaces. These are filamentous algae. These would be things like Polysiphonias, Cladophorus, and so on. These are sheet-like algae, which would be like ulvas and enteromorphas. And these are microfilamentous algae. These are like colonial diatoms and small blue-green colonies. So if you compare the 70s and the 2000s, let's do this first at the more urban sites. You can see at Corona del Mar a drop in fleshy and an increase in articulated corallines and an increase in crustose. Now, we have some changes in these others, but these are the ones I want to emphasize. So this is pretty similar to what we saw by the resurveys of Dawson sites. If you go to White's Point, this other urban site, you can also see an increase in articulated corallines, a small decrease in fleshy. There weren't a lot of fleshy there to begin with. Crusts were dropped a bit, but also this same sort of urban signature of really low cover of fleshies and high cover of articulated corallines there. Less or fewer differences were appearing at the sites on the outer edge of urbanization. At Dana Point, we actually see an increase in fleshy algae, a decrease in articulated corallines, and our larger seaweeds stayed about the same. At Paradise Cove, the larger seaweeds were really a big, abundant part of the flora. Fleshies, a little bit more so in the 70s. Articulated corallines, a slight increase, and so on. So it looks like, at least at Corona del Mar, we got some things going on here, and probably at White's Point, and a little more stasis, if you will, in the two outer urban sites, based upon looking at the relative abundances or the cover that's being generated by these form groups. Okay, so we concluded from this work that on these mainland shores, most of the sites showed changes in flora and fauna and substrate topography. I might point out here something interesting, and that is that we actually replicated the exact plots, or as close as we could to the exact plots that were taken in the 70s. We replicated those and resampled them in the 2000s. And we were able to locate those in most cases, but we did find that in most of these sites, the substratum had changed. So there was movement of rocks, which means that going back to the same exact plots has a little bit of a limited value. We found that different populations, so this is going down looking at the species, different ones changed abundances at different sites. And in our more urban sites, we found that generally the larger fleshy species of the larger fleshy algae, which you see represented up here, declined. And we found that cover tended to be more dominated by these crusts and articulated corallines and some of these small turf-forming algae. Now, so the questions that come to mind and what we entered this work thinking about that we did in the 2000 period was what might be the causes of some of these changes? First, we had to document them. And what are the ecological implications? Now, two things I want to say here. First off, different things are happening at different sites. So it's very hard to come up with trends that are going to be clear and concrete across all these sites. That makes it more difficult to interpret the data. The second thing I'd like to say is that so many things are going on and went on in the Southern California area, changes in ocean conditions, El Ninos, urban influences of runoff, different chemicals that are put into the environment, the nearshore environment, as a result of manufacturing efforts, changes in sewage treatment, influxes of people that come in and collect and trample over intertidal surfaces. That sorting out the causes seems to be pretty much an impossible task. But actually, what I think is even more important is to focus on what the ecological implications might be of some of these changes. So the last part of this talk, we're going to talk a little bit about this. So the first thing I want to say is that if indeed there is a decrease in a lot of these larger algae and an increase in some of the smaller ones, what does this have with regard to impacts on habitat provision for invertebrates? Because certainly larger invertebrates are going to largely be found under some of these larger canopies in the intertidal zone, particularly when the tide goes out and they seek refuge from desiccation and from temperature increases. And when you get into a flora dominated by smaller seaweeds, well, you might expect to see smaller invertebrates. This is something that's worth pursuing. We didn't do this or look at this in detail, but this may well be a trend that has occurred, because certainly when you go back and look at museum collections, there are a lot fewer specimens that we're finding now of larger, particularly shelled macroinvertebrates on our rocky intertidal shores that are available to be found during low tide periods. Okay, I'm going to talk a little bit about another master's thesis, this one done by Amy Bullard in 2005. It's a master's thesis that she did. It's in the Cal State Fullerton library. And what Amy did was to come in and look at abundances of seaweeds using point contact procedures, 5 transect lines to spread this area out, and percent cover data was generated from this, as well as a list of species that were present over this intertidal area. And then she did light saturated primary productivity of the seaweeds that were present on all these that she found at these sites using oxygen methods, converting the oxygen to milligrams carbon and expressing the data on a per area, seaweed area basis, so that she could use the cover data and the productivity area data to generate productivity values for the sites. So here we're investigating primary productivity of these communities. Now, it's been known for a long time that there is a pretty strong relationship between the primary productivity, the net primary productivity, light saturated primary productivity of a seaweed and its morphological form. Crustose algae have pretty low net primary productivities. This is a summation of a bunch of work that was done to generate these numbers. And the data here are expressed as milligrams carbon per gram dry weight per hour. So it's a rate. And you can see that some of the larger productivities, if we look at this, these thin sheet-like algae, where pretty much every cell is exposed to the light and is photosynthetically active, pretty high productivities per area. And that our kelps and fucoids have pretty high values, certainly compared to crustose and compared to articulated corallines. And these fleshy algae, particularly those that are branched, also have pretty high values. So this would mean that a seaweed flora dominated by seaweeds that are in this group would be expected to have higher productivities than those dominated by species in this group. Now, Corona del Mar was one of the sites that Amy Bullard looked at. And this was a site that had a pretty long record of previous work. All of these sites were looked at before. And this is Corona del Mar. And then Dana Point, remember, this is that more on the outer urban edge. Hadn't been a lot of ecological work done there, but this was a Dana Point site. So this is the results for cover. And you can see that winter and summer, 2 seasons were assessed. Corona del Mar had a mean seaweed cover of about 70% and relatively, well, little difference, if any, between winter and summer. Dana Point, on the other hand, had a little higher cover value and a pretty big difference between summer and winter. And this isn't unexpected. In Southern California, winter low tide periods are pretty harsh, and a lot of the fleshy algae tend to die back a bit, and the cover drops. Whereas in summer, longer days, more benign intertidal conditions, and those same algae tend to be on growth spurts, and they tend to grow and become more abundant. Now, you'll notice that there's a significant difference between the 2 sites. Season was not significantly different, but there was pretty much a close to significant difference as an interaction between site and season, largely driven by this particular increase at Dana Point. Now, if you look at the net community primary productivities, this is averaging up in milligrams carbon per meter squared per substratum per hour. You see that, again, we have a significant difference between the 2 sites. We have no difference in season and no interaction. So based on just simply the overall net community productivities, sites are different, but seasons not so. On the other hand, if you look at what seaweed species were generating the productivity, you get a different result. So each of these points on these 2 graphs represent one of the 5 transect lines. As before, we have summer here in the dark green, and winter in the light green at Dana Point, and summer here in the orange at Corona del Mar, and winter here. This is the cover plot, and you can kind of see that a little bit of movement of Dana Point in this direction towards Corona del Mar in the winter, but nothing really to write home about. But the 2 sites have definitely different cover patterns when looked at this way. So I want to point out that this looks at each individual seaweed species, its cover, and looks at the whole community in that light to produce this non-metric multidimensional scaling plot. Now, if you look at the productivity plot, you get a big different result, because here, the productivity profiles, I'm calling them, at Dana Point in the summer are really a lot different than during the winter. This means that different species are contributing to these productivities in the summer than they are in the winter. In other words, a large fleshy seaweed may be contributing a lot of the productivity in the summer and not as much in the winter. Whereas in the winter, the articulated corallines may be contributing more of the productivity than they do in the summer. And that creates this big difference. At Corona del Mar, not a big difference at all. So again, sites, big difference, but here, season is quite different. So we interpret this as meaning that in the winter, when conditions were harsher, low tide conditions were more rigorous, that Dana Point was being dominated more in terms of the productivity by more disturbance-resistant species types, which include crusts, articulated corallines, and less so by the more longer-term, perennially present, larger fleshy seaweeds. It was beginning to look more like Corona del Mar. So this is a sort of shows you that result, again, by looking at the contribution of site productivity at the two sites and looking at these different groups. So right away, you can see that at Dana Point, fleshy algae were making up a whole lot of the productivity for the site, particularly in the summer but also in the winter. Whereas at Corona del Mar, much less. Articulated corallines were much more important contributors to productivity at Corona del Mar in both seasons, but much less so at Dana Point. So this tells us that these sites have different productivity profiles, and that has some potential implications. So the results of this work, comparing Corona del Mar versus Dana Point, we would find the total site productivities, they're greater at Dana Point, but the profiles differ. That means the contributions by the species types differ. And the winter productivity profile at Dana Point tends to resemble Corona del Mar. So in the end game, when we look at this as an implication, there's some additional questions that follow. What does this have to do with energy flow in these systems, and what are the food web implications? So that's what I got, and think I've got a little time here to answer any questions. So this is what I've tried to share with you today, and I know that it goes back maybe to a period before some of you were even born, but historical work is important in a lot of ways, because it puts in context what you're doing today and what you're finding today. And it gives you some indication that things are indeed changing. And we are in a period of rapidly changing ocean conditions. We should expect to see changes occurring along the Southern California and the California coast and elsewhere throughout the Pacific coast of North America. So thanks for your attention. And I hope this has been a little valuable for you, and I'll be happy to try to answer any questions that any of you might have.